Hi everyone, and welcome back. Today I want to present a new method that I genuinely find exciting, a new way of representing protein sequences called protein sequence extended connectivity fingerprints. At first glance, it might seem like just another encoding strategy, but it is much more than that. Think about it this way. Proteins carry out the work encoded by DNA, and turning their long amino acid sequences into something a machine can understand is difficult and often computationally expensive. Protein sequences can be represented using various methods, from lightweight encodings such as one-hot encoding to deep learning-based representations generated by large protein language models. Each is useful, but each has limitations. You either lose important biological context or you pay the price of huge computational cost. And that's why this new method feels refreshing. It borrows the elegant idea behind extended connectivity fingerprints, a technique that has transformed how small molecules are represented. But here, the algorithm walks along the protein sequence, capturing physicochemical properties, local neighborhoods, and contextual information, and compresses all this information into compact, machine-friendly, fixed-length fingerprint vectors. What I like about this approach is its balance. It captures biologically meaningful information while remaining lightweight, interpretable, and computationally efficient. You can actually use it in large-scale protein modeling workflows without needing giant embeddings, structural data, or expensive GPUs. And the results are not just theoretical. The frequency-aware variant of these fingerprints has reached competitive and sometimes superior accuracy in predicting the pathogenicity of missense mutations, a task where many classical encoding methods struggle. That means this fingerprint doesn't just compress information, it captures biology in a way that still matters, even for difficult real-world predictions. To me, this is what makes the method really valuable. We get state-of-the-art biological signal without the overhead that slows down discovery. So, over the next few minutes, let's explore how these fingerprints work, why they're meaningful, and how they can open new possibilities in bioinformatics, drug discovery, genomics, and protein engineering. Before we discuss this new method, let's briefly revisit some common strategies currently used to represent protein sequences for machine learning. One category involves simple discrete encodings with methods like one-hot encodings and K-mers. Such approaches convert amino acid sequences into binary or frequency matrices, capturing residue identity or short local fragments along the sequence. They are easy to compute and widely used, but they tend to struggle with long-range interactions and higher-order context. Another class of techniques relies on physicochemical descriptors. Here, each amino acid is mapped to numerical properties such as hydrophobicity, charge, volume, or flexibility. These descriptors improve biological relevance and can model structural or functional trends, but they still require careful engineering and may overlook complex contextual relationships in the sequence. Another category of methods learn rich contextual embeddings by training on millions of sequences, allowing them to capture subtle evolutionary constraints and long-range dependencies. As a result, they often deliver excellent predictive accuracy across a wide range of protein modeling tasks. However, generating these embeddings can be computationally expensive and harder to scale for large datasets or real-time applications. In summary, each representation category has clear strengths and limitations, and choosing the right one depends on the balance between predictive accuracy, interpretability, and computational cost. Now that we've seen a few examples of how protein sequences can be represented, let's look at how protein sequence extended connectivity fingerprints was developed. The idea behind comes from a well-established method in chemoinformatics known as extended connectivity fingerprints, which is often used to describe small molecules. The authors adapted this idea to proteins by treating each amino acid like a node in the linear graph and expanding its neighborhood along the sequence, instead of around atoms, as in classical small molecule fingerprints. To build the fingerprint, the method begins by representing each amino acid as an array of normalized numerical values that reflect selected physicochemical descriptors. These descriptors, collected from curated biochemical databases, ensure that each residue is encoded in a biologically meaningful way rather than simply as a symbolic letter. Next, the algorithm iteratively expands the sequence neighborhood around each amino acid. At each iteration, it incorporates the identities and physicochemical descriptors of neighboring residues, as well as their positional distance from the focal residue. This means the representation encodes not just the properties of a single amino acid, but also the local context in which it appears along the sequence. As the expansion radius increases, the method progressively captures longer-range dependencies and recurring motifs that may be relevant for structure or function. Each of these contextual fragments is then hashed into a fixed-length vector, producing a compact numerical fingerprint for the entire protein. The authors propose two types of representations. A binary method, where each position in the final fingerprint is either 0 or 1, and the count method, or frequency-aware version, which can capture how common or rare specific motifs are, which often leads to better predictive performance. Overall, the protein sequence extended connectivity fingerprints method offers a balance between biological knowledge and computational efficiency. When evaluated on large Missense mutation datasets, the count-based version of the method consistently outperformed or matched advanced sequence descriptors and achieved state-of-the-art predictive scores, while being orders of magnitude faster to compute than large protein language model embeddings. To keep this video short, I won't discuss the application of the method further here, but I encourage you to check out the papers, as the findings are of considerable interest. Here you can see a screenshot of the GitHub repository, and I've also added the URL in the video description below. The authors provide clear instructions on how to install the tool and how to run it. On my side, I installed it on a Mac using a slightly simplified protocol, as shown on the screen. The installation was straightforward, and the tool runs smoothly. It is indeed fast in generating the fingerprints, even when processing multiple sequences. This brings us to the end of today's presentation. I hope this elegant and efficient package will be useful for your own research projects and help you explore protein sequences more effectively. Thank you for watching, and until next time, take care.